And let me introduce Dr. James. It is my absolute honor and privilege to welcome a truly distinguished guest today, Dr. James D. Rosenbaum. Dr. Rosenbaum is a physician scientist whose contributions to both rheumatology and ophthalmology have not only advanced the medical research, but also had a profound impact on countless lives. As many of you know, Dr. Rosenbaum has held esteemed positions such as Senior Vice President for Research at Corvus Pharmaceuticals and Chief of Ophthalmology Emeritus at the Legacy Devers Eye Institute. His pioneer work in ophthalmology, microbiome, and rheumatology, including over 600 scholarly publications, has shaped the very way we understand the intersection of rheumatology and ophthalmology. And let's not forget that he is the only rheumatologist to have ever led an ophthalmology department, a remarkable achievement that speaks to his broad expertise and vision. Dr. Rosenbaum, your contributions to Proton and your continued support has been truly invaluable. From the moment you lent your expertise and insight to our work, you have inspired us to reach new heights in our shared mission. On behalf of everyone here at Proton, I want to extend a heartfelt thank you for your unwavering commitment, guidance, and support. It is because of thought leaders like you that we are able to drive innovation and make strides towards a better and healthier future. So please join me in welcoming Dr. James T. Rosenbaum. The stage is all yours.
William, that's very, very kind introduction. Thank you very much. But I'm going to show you how stupid I am in the next 40 minutes or so, because I've been trying to solve a problem since 1978, which is 46 years by my calculation. And I guess I get an A for persistence, but I don't get an A for solving the problem because I think another group really gets most of the credit for advancing how we understand the problem. So the problem is, how does the HLA-B27 molecule predispose to disease? That's the riddle, that's the puzzle. And that, of course, B27 relates to ankylosing spondylitis, reactive arthritis, and acute anterior uveitis. So if I can advance, I'm going to first describe the puzzle. And then I'm going to seg away from the puzzle and give you background about the microbiome. I'm going to explain how I became interested in the microbiome and how bacterial products could cause inflammation. And then I'm going to finally, hopefully, solve or at least advance the answer to this whole issue. So I was very fortunate to do my postdoctoral fellowship in rheumatology with Hugh McDevitt. And Hugh McDevitt, along with a fellow named Baruch Benacerraf, those two were the fathers of immune response genes. They were the first people in the world to realize that the immune response is under genetic control. And McDevitt and Benacerraf wrote a commentary in Science 52 years ago. And they said, hey, look, we have genes that influence our immune response. Those are the major histocompatibility complex genes, which we call H2 in a mouse and we call HLA in people. And we have a whole bunch of diseases like rheumatoid arthritis and systemic lupus that are caused by the immune response as best we know. So it's our prediction that diseases like rheumatoid arthritis and lupus will be influenced by one's HLA type. And it only took about a year for that hypothesis to be confirmed, but the hypothesis was confirmed in a truly unanticipated way. It was confirmed by a group in London. They published in The Lancet, and a group from UCLA who published the very same month in the New England Journal of Medicine. They both came to the same conclusion. And the conclusion was that an HLA type that was then called HLA-27 and subsequently became known as HLA-B27, that HLA type didn't predispose to rheumatoid arthritis. It didn't predispose to gout, which is a disease they studied because gout was clearly genetic. It predisposed to the control in the experiment. The control was a disease called ankylosing spondylitis, which the scientists at the time thought was neither genetic nor immunologically mediated. But it clearly is immunologically mediated, and it clearly is genetic. And the main genetic factor is HLA-B27. And as I said, it predisposes not only to ankylosing spondylitis, and you see the progression here from a famous set of photographs, someone joining the army in Canada, and it predisposes to acute anterior uveitis. So the McDevitt-Benacerraf hypothesis was way ahead of its time. If you spend several $1 million and you do a genome-wide association study, a GWAS, you might identify 100 genes that predispose to ankylosing spondylitis, but almost always the major gene in an immune-mediated disease is coming from the HLA complex. It's usually a major compatibility complex gene that influences getting an immune-mediated disease more than any other gene, although all of these diseases are complex immunologic diseases for which there are very many genes that do influence it. We know an incredible amount about B27, okay? It turns out that there is no genetic system in the body that's as diverse or polymorphic as the HLA system. If we just take the HLA-B locus, there are more than 700 B alleles. And even within B27, there are way more than 100 different alleles or subtypes. B2701, B2705 is the most common. Of all those subtypes, it seems like every single one predisposes to ankylosing spondylitis with two exceptions. One is B2706, and the other is B2709. But otherwise, everyone predisposes to disease. And here you see the three-dimensional structure of B27. Again, we know a tremendous amount about this predisposing molecule. So even though we know so much about it, and we know that there are more than 100 diseases whose susceptibility is influenced by HLA, we don't really know so much about the mechanism. So McDevitt and Benacerraf said, well, the mechanism would be it's controlling the immune response. But is that true? It's been very hard to prove that. And so my colleagues and I published a commentary on the 50th anniversary of the Benacerraf-McDevitt paper. And we said, you know, again, we know that HLA controls the immune response, but we have very little evidence that HLA molecules control the immune response to a self-antigen, an autoantigen. And so maybe what's going on is that the HLA molecules are controlling the immune response to a bacterial antigen. And I'm going to talk in a few moments about those bacterial antigens. Just parenthetically, one disease where we do think that HLA molecules control the immune response to the triggering antigen is celiac disease, or SPRU, where the antigen is gliadin. But that's, remember, not a self-antigen. That's not an autoimmune disease. Gluten is a foreign antigen. The other possible exception might be HLA-DR4 and cyclic citrullonated peptides and rheumatoid arthritis, but it's probably the one exception out of the 100+ diseases that confounds the rule. So in terms of B27, a friend of mine named Joel Torog summarized a symposium that met in 2010, and they offered 3 reasons to try to explain the mechanism. And Reason number one was that B27 controls the immune response to an autoantigen. But again, we hadn't found that autoantigen. We still haven't found, or maybe we've found that autoantigen. We'll see. B27 is different from other HLA molecules in that it tends to form a dimer on the cell's surface, and that can activate natural killer cells. And B27 is also unstable within the cell, and it stimulates a series of garbage collection type responses within the cell that are called the UPR, the unfolded protein response. And Bob Colbert, who's now at the NIH, has written a fair amount about that. But it's easier to show the importance of the unfolded protein response in a rat than it is in a person. So who knows? So that's the puzzle. I'm going to leave the puzzle and just make sure that we're all sort of on common ground in terms of the microbiome. And on this slide, you see kind of a crude picture, right? And I think you'll recognize that this is a drawing of a sperm penetrating and fertilizing an ovum. And it's not a great drawing, you might say, but it turns out it's a prize-winning drawing. It's a prize-winning drawing because of who the artist is, or I should say, who the artists are. The artists are bacteria. This was drawn by bacteria that were plated on a petri dish with different substrates in different regions of the petri dish. And then the bacteria released enzymes to cause colorimetric reactions that caused this image. And so the American Society of Microbiology holds an annual contest to see what bacteria can draw, and it gave first prize a few years ago to this drawing. So the theme here is that bacteria do some very surprising things, and we need those bacteria. Bacteria in our body outnumber the cells in our body. We used to say 10 to 1. It's probably more closer to 1.4 to 1. If we don't look at the number of bacteria, but instead we look at the number of RNA transcripts in the body, for every human transcript that Rupesh's body expresses, there are about 100 bacterial transcripts that are being expressed. So on that basis, the bacteria, Rupesh is about 1% Rupesh and 99% bacteria. We have only started to appreciate this bacterial world recently, and I'll get into that in a moment. But most of the bugs that live on us or in us or around us, especially in us, are anaerobic, and therefore they're very hard to culture. But we rely on those bacteria. I argue that those bacteria are the main way that our immune system gets educated. They produce neurotransmitters like serotonin. They are the major source of our vitamin K. They're very important to our health, although they can rarely cause disease as well. So in 2007, the NIH started to fund what's called the HMP, the Human Microbiome Project, and a similar project got started in Europe called MetaHit. And in both cases, both projects were basically a catalog to understand what these bacterial microorganisms are. And every few years, the HMP and the MetaHit would announce their results. And whenever they would do so, it would make the cover usually of both Nature and Science at the same time. It's really been a very productive project. And the reason it got started in 2007 has to do with money. It has to do with cost. So this is a slide. Craig Venter, who owned the biotech company called Celera, decided hey, wouldn't it be nice to know every nucleotide in my genome, every single one of the 3.1 billion bases in my genome? And he set about to do that. He was racing the NIH. It cost him in 2001 $100 million, but he more or less sequenced his entire genome. The cost of doing that has fallen precipitously, so that now it's around $1,000 to sequence your entire genome. Moore's law, is the chip law. I'm just going to take a sip of water. So Moore's law is the law about how we've had an exponential increase in the capacity of microchips, and the cost of genome sequencing has changed even more dramatically than the expansion of memory in microchips, as predicted by Gordon Moore. So my first dawning of the importance of the microbiome was in 2010. And it came from a paper that appeared in Science about the metabolic syndrome in mice that lack Toll5. So I think that every one of you probably knows that we have an immune system that we call our adaptive immune system that's very tailored, very specific, and an innate immune system that is much less discriminating, but faster to respond. And the innate immune system relies on a variety of receptors, some of which are called tolls, one of which is Toll5, which recognizes flagellin, which is a bacterial protein only expressed by certain bacteria which have a flagella. So if you delete Toll5 from a mouse, you create a Toll5 knockout. Not surprisingly, you change the bacteria that are in the gut. What wasn't anticipated when you create a Toll5 knockout is that the mice would get fat and they would become insulin resistant, and basically they would have metabolic syndrome or prediabetes. But that's not why this paper was in Science. The paper was in Science because the metabolic syndrome was a contagious disease. And they showed that by taking these Toll5 knockout mice and co-housing them with germ-free mice that were genetically identical, but they had a normal, except for having a normal Toll5. So they had the same background, which I think was C57 Black 6 for them, not actually certain at this time, but they were the same mice, but these mice had been delivered by cesarean section so that they came out without exposure to bacteria. They were placed in a cage with a bubble on top to prevent bacteria from entering through the air, and all the food was sterilized. So as a result, the mice never developed a normal immune system. Their guts never became colonized, but mice are coprophagic. They eat each other's poop, and in so doing, the germ-free mice acquired the microbiome of the Toll5 knockouts. And they didn't get quite as fat as the Toll5 knockouts, but they got somewhat fat, and they got insulin resistant. They got metabolic syndrome. In other words, the microbiome played a major role in the metabolic syndrome in mice. So you might be saying, well, that's a nice mouse experiment, but it has no relevance to people, except there's a scientist in St. Louis in the United States. His name is Jeffrey Gordon. He's the senior author on both these papers. He's been studying people who are starving in Africa and people who are starving in Nepal. In Africa, they have a disease called kwashiorkor. And he studies identical twins, where one twin is dying from weight loss and the other twin is not dying. And he shows that their microbiome in their gut is different, that if he takes the microbiome from the twin dying of starvation and gives it to a mouse, the mouse begins to lose weight. And he's developed a prebiotic, a diet that changes the microbiome and helps to prevent weight loss. And he's shown that in both a part of Africa as well as in Nepal. It's really brilliant work. So I don't have time to show you all the incredible experiments that have been done and are being done to show how important the microbiome is in changing the immune system. But as I said, if you raise a germ-free mouse and you don't let the mouse's gut get colonized, that mouse has shriveled lymph nodes, shriveled spleen, and a real inability to mount a decent immune response. So I am going to now leave the microbiome partially to tell you how I got especially interested in the microbiome, and that has to do with how important bacterial products can be in inflammation. So I joined the McDevitt lab in 1978. That's when I started my fellowship. It was just 6 years after the McDevitt Benacerraf essay. It was 5 years after the B27 relationship had been realized. And I wanted to develop an animal model of reactive arthritis. And in people, reactive arthritis can be triggered by Shigella, Salmonella, Yersinia, Campylobacter. And so I grew up those kinds of bacteria in broth. I killed the bacteria, and then I injected them into the footpad of rats, thinking that the rats might develop a reactive arthritis. And to my disappointment, they didn't. But they universally developed an acute anterior uveitis. They developed red eyes and irritated eyes. And it was clear that gram-negative bacteria did that, gram-positive didn't. And you could just take the lipid A moiety from the lipopolysaccharide or endotoxin, inject that subcutaneously or intraperitoneally or into the footpad, and within 24 hours, the eyes would become inflamed. And it's really because of that fortuitous observation that I, as a rheumatologist, became clinically interested in anterior uveitis. So I mentioned Torog and the essay he wrote in 2010 summarizing that conference and the speculation as to why did B27 predispose to disease. Torog created rats that expressed the human HLA-B27 and the human light chain that helps B27 appear on the surface of a cell, beta-2 microglobulin. And those transgenic rats get dactylitis, like people with B27-related disease, and they get a spinal arthritis. Here you see what happens to their tail, which is analogous to the spine or continuous with the spine. Torog did an experiment in 1994 raising these rats germ-free, delivering them by C-section, sterilizing their food, and guess what? They did not get the gut disease that they get, and they didn't get nearly as much joint inflammation. So there's another model now in mice of spondyloarthropathy. It's called the SKG model or the ZAP70 model with a mutation in ZAP70. It needs a fungal antigen to kind of trigger it. But again, if they're raised, these are mice now, not rats. if they're raised germ-free, they're protected from the disease to a large extent. So my colleagues and I summarized some of our observations about 2011 or so, and published an essay saying, hey, look, reactive arthritis, which is a B27 disease, is related to bacterial infection. If you get Crohn's or ulcerative colitis, you can get sacroiliitis, peripheral arthritis, and uveitis, just like the B27 disease ankylosing spondylitis. The germ-free rats and germ-free mice are protected from developing spondyloarthritis. And sulfasalazine is an antibiotic that's designed to stay within the gut. It reduces your gut bacteria from trillions. It reduces it by 99%, which means you get down into the billions. You still have plenty of bacteria, but it also helps to treat ankylosing spondylitis, at least the peripheral arthritis, and it helps to prevent the attacks of acute anterior uveitis. So we thought that maybe the microbiome or the bacteria in the gut could be the key to understanding spondyloarthritis. But we were largely ignorant of a literature that was 100+ years old that also supported this idea. And one of the pioneers in this field was an ophthalmologist whose name is A.C. Woods. A.C. Woods was a very prominent ophthalmologist. He was the founder of the Wilmer Eye Institute at Johns Hopkins. And some people would say that that's the top eye institute in the world. So Woods also grew up gram-negative bacteria in broth, and he would spin it down, and he would inject just the broth into rabbits intravenously. And he showed that the eyes developed a round cell, that's code for a leukocytic, infiltrate around the ciliary processes. So he was inducing an acute anterior uveitis in rabbits. In the 1920s and the 1930s, we had an impoverished therapeutic armamentarium to treat any kind of inflammation. My dad was a rheumatologist who started his practice in the late 1940s. And basically, he treated rheumatoid arthritis with aspirin and with bed rest. This is a paper published in 1932. It came from the Mayo Clinic in Rochester, and it was presented at the first ARVO, or the second ARVO conference, which was held in Philadelphia in 1931. And it talks about how uveitis was treated at the Mayo Clinic. And the way it was treated was to look for occult foci of infection. And those foci were usually teeth, tonsils, prostate, or cervix. And if you had uveitis and you were referred to the Mayo Clinic, they would treat you surgically by doing things like removing all your teeth or drilling your sinuses, or if you were a woman, you'd probably have a hysterectomy. That was the state of the art in the 1930s. They were onto something about bacteria, but I don't think their treatment was very effective. But again, this wasn't just in ophthalmology, it was also a way to treat rheumatoid arthritis. So one of the first questions we asked in the lab is, if our hypothesis is correct, HLA molecules should have some influence on the gut microbiome. And we did a study in collaboration with Matt Brown, who's in London. And the study in my group was led by Mark Asquith, who was an assistant professor and my collaborator. And we generated data on both ankylosing spondylitis with B27 and rheumatoid arthritis and DR4. And we showed convincingly, we felt, that those particular HLA antigens influenced the bacteria, which bacteria were present in the gut microbiome. And we published that in A&R 5 years ago. And just parenthetically, I should say that Mark passed away about 4 years ago from a cerebral aneurysm, and he was a genius. I would have more information to share with you if Mark were still alive. We also did a similar study on A29, which, as you should know, predisposes to birdshot retinal choroidopathy, and showed that A29 has an effect on the gut microbiome. I also want to do one other tangent. I know that some of you are aware of this very clever essay that was published in PLOS One in 2006. And the first author was Dennis McGonigal. And Dennis tried to put immune-mediated diseases on a spectrum. He said that some diseases are autoimmune, and he found genetic diseases like APSED, which indeed have to do with gene mutations and are autoimmune. And he found other genetic diseases which he called autoinflammatory, and others have called autoinflammatory. And these diseases involve usually mutations in our innate immune system. And both the genetic diseases like Blau syndrome that causes inflammation in eyes and joints, and ALPS or IPEX or APSED, which are autoimmune diseases, these are all rare diseases. But if you take more common things like rheumatoid arthritis, ankylosing spondylitis, psoriatic arthritis, he put them on a spectrum where he thought, hey, some could be an autoinflammatory component, an autoimmune component, and some, like spondylitis, he thought would have both. And also, I apologize for going on these tangents, but this essay didn't consider the possibility that the immune system could be causing diseases in which the antigen was completely foreign, such as sprue. So there's another dimension that McGonigal didn't consider, but nonetheless, it was a very thought-provoking essay that still has information for us today. Okay, I've set the stage, I hope. Now I'm going to show you or share with you the paper which I think advanced the field dramatically. And that was a paper that was published 2 years ago this December in Nature. And it came from international groups. There was a group at Stanford. Chris Garcia was the senior author leading the group. And it involved a collaboration with a couple of ophthalmologists at WashU St. Louis, Paley and Hassman. Actually, Paley's a rheumatologist, Hassman's the ophthalmologist. And it came from a group in Moscow that had been collecting synovial fluid from patients with spondyloarthritis. The group at WashU St. Louis was collecting aqueous humor from patients with B27-associated acute anterior uveitis. And Garcia analyzed the peptides that were found in the B27 sitting on the lymphocytes, either invading the joints or invading the eye. And he showed that similar peptides could be eluded from either the synovial lymphocytes or the ocular lymphocytes. So he analyzed these peptides. He generated libraries of peptides with a yeast library and analyzed what would bind. He showed with using single-cell RNA-seq that there was an oligoclonal expansion of T cells that had the T cell receptor TRBV9. And if you have an oligoclonal expansion of T cells, it implies that it's an antigen-driven process. And using that peptide library, he was able to identify several autoantigens, one of which was called GPER1 or G protein coupled estrogen receptor 1, which is expressed by the iris and ciliary body. He was able to identify some self-antigens, autoantigens, in the B27 binding group. Really seminal observation. So it is an autoimmune disease. But the catch is that it's also a microbiome-driven disease because he also identified a variety of bacterial antigens, one of which was called YEIH, that turns out to be expressed by multiple different gram-negative bacteria, including a variety of gram-negative bacteria that have been associated with ankylosing spondylitis. There was a subsequent paper from the Moscow group that appeared in Nature Medicine, and they showed that they treated a patient with ankylosing spondylitis who was not getting better with antibodies to TNF, and they instead gave antibodies to this BV9, and got the patients better as a result. Really had a significant clinical benefit by deleting the lymphocytes that were expressing this one particular receptor. And of course, this had targeted immunosuppression, so it wasn't broad immunosuppression. There was very little immunocompromise by doing this. That's why it made Nature Medicine. So I do think that this was a seminal advance, although like all good science, when you learn something, it's just a platform for asking more questions. One question is, which comes first, the immune response to the microbe or the immune response to the autoantigen? I think it's more likely that tolerance is broken by the bacteria, and then you get an autoimmune response. And there's precedent for that, for example, in Guillain-Barré syndrome, or in rheumatic fever. We also need to explain, why doesn't everyone with B27 get spondylitis? It's only a minority. And we need to look at the first cousin diseases like psoriatic arthritis, the arthritis with ulcerative colitis and Crohn's disease. Are those targeting the same antigens, or are they different? And if indeed this is an autoimmune disease, shouldn't it be more consistent? Why should it be episodic? So there are lots of things yet to do for the people who are William and Carlos's age and still have hopefully 50 more years to contribute to science and try to attack these riddles. So I have a couple more slides. I'm going to take a break for just a minute, and then I don't know whether you want me to answer questions or walk through another couple of slides. Rupesh, what would you like?
I think, yeah, you can complete this couple of slides then, Sunny. What do you think, Sunny? You should.
Yeah, I'd love to follow what he has.
Okay, give me just a moment and I'll resume, okay?
Yeah, sure. So in the meantime,
are you asking a question? Or just
no, the Nature paper is
the Nature paper, and you agree with me it was seminal, I think. Yeah. I'm going to have to redo my lecture pretty soon because it's going to be too long since that Nature paper was published. And but when I talk to ophthalmologists, usually they don't know the paper. So that's for my good fortune. This is a paper that I wrote with Mark Asquith in 2018 in Nature Reviews Rheumatology. And Mark and I suggested 4 different hypothetical reasons why the microbiome might cause disease. And I think it's possible that all 4 contribute. But there's evidence, for example, that lymphocytes get educated in the gut and migrate to other organs like joints and eyes. There is lots of evidence that the microbiome organisms will influence how many IL-17 positive cells you have, Th17 cells, how many Tregs you have. They have a profound effect, as I said, on the immune system. We thought that mimicry could easily be a contributor, and we showed it here, and the Nature paper goes along with that. And then it could just be that there are things like endotoxin, that if you change the microbiome, you could change how permeable the gut is. And the bacterial products are potentially phlogistic or inflammatory, including peptidoglycan, which is in both gram-positive and gram-negative cell wall, and endotoxin especially. And we've shown, and haven't yet published, that the amount of endotoxin in someone with ankylosing spondylitis increases in the blood. And that's been shown in inflammatory bowel disease. Anat Galor is an ophthalmologist at the Bascom Palmer in Miami, and she published last year a collection of essays about the microbiome and ophthalmology. And we contributed a chapter and this diagram, and I don't want to belabor this diagram, but anything that you ingest is going to be affected by the microbiome. So I think many of us who treat uveitis use methotrexate, for example. It's very clear that methotrexate is metabolized by the microbiome. Paracetamol, which we call acetaminophen here in the US, is metabolized by the microbiome. 100% of my patients ask me about diet and what I can eat to prevent. And it's very clear that just as different plants grow where it's wet as opposed to the desert, different bacteria grow depending on how much sugar, how much fat, how much roughage, how much, your diet clearly dictates what bacteria will thrive within your body. I talked about HLA. I talked maybe a little bit about dysbiosis, immune education. My hope would be that someday we will understand this enough that we will prevent the disease. And already we're using it for therapy, as in fecal microbiome transplants for C. diff infections. But we're going to be more sophisticated compared to that crude technique. And then one fascinating strategy has been to use a prebiotic to try to induce what has become arguably the best-selling group of drugs in the world, these GLP-1 drugs that cause weight loss. And so a predominant bug in our gut is Akkermansia muciniphila. Muciniphila because it depends on the mucus in our gut. It really is very abundant in the gut. And I'm not endorsing Pendulum, and their peer-reviewed data is not very cogent. But their idea is that Akkermansia will induce the body to produce its own GLP-1 and be a less expensive approach than the Ozempic, Wegovy, Zepbound approach. But this is why science has talked about the promise of poop. It really does have a lot of potential, I think, in our understanding of disease and therapeutic armamentarium. So that's the end of the talk. I hope what I've convinced you is that the pathogenesis of both ankylosing spondylitis and acute anterior uveitis is probably an example of mimicry, where it's a B27 presented microbial antigen and a reactivity to a self-antigen. But there are lots of questions that remain, and maybe one day we will build on the Nature paper and induce tolerance to these self-antigens, and we will only be able to read about ankylosing spondylitis. It'll be a disease like rheumatic fever that's sort of disappearing. That's my hope. So thank you for the opportunity to share these thoughts.
Thank you very much, Dr. Rosenbaum, for this amazing talk. I think there are many questions now, and we have some minutes. I will take 30 seconds to introduce Dr. Sonny, who will serve as a moderator. Dr. Sonny Wong, he's a clinician scientist and associate professor at the Lee Kong Chian School of Medicine at NTU in Singapore. He received his MB, his Bachelor in Medicine and Surgery with honors at the Chinese University of Hong Kong, and his PhD in University of Oxford on human genetics and infectious disease. He completed his clinical training in gastroenterology and endoscopy in the Prince of Wales Hospital in Hong Kong. His main research interest is on gut microbiome, investigating the host microbiome interaction in digestive and metabolic diseases and exploring for for discovery of novel biomarkers and therapeutics. He has won several awards, including the Asia Pacific Digestive Week Emerging Leader Lectureship, the Sir David Todd Lectureship, the Lojin Sekshi Wei Foundation Emeritus Research Award, and the Korshor Foundation Award. He has published over 160 peer-reviewed articles, including papers in Nature Genetics, Nature Communications, Gastroenterology, and Gut, and he's currently an associate editor for the Journal of Gastroenterology and Hepatology. And the most important for us is he's a member of Proton, and he's actively contributing in the team. Thank you, Dr. Sonny, for joining us.
Yeah, thank you, William. I think that's too long the introduction for modulator, and but thank you very much, and thank you for James. It's very insightful talk, and so I maybe I would ask the floor if there's any question. Or else I do have one that James, you mentioned about the diet and nutrition in one slide, and I think we have talked about it, and there's huge interest. Where do you think the role is the diet on? Would that be true actually, because the diet itself may have effect on the host and also the immune system, and also of course it will gear the microbiome as well. How do you see the role of diet in autoimmune diseases?
Yeah, so it's a wonderful question. And as you said, you and I have had a chance to discuss it some previously. The microbiome is obviously very complex, and our diet is very complex. So I don't know that the pepper that I put on my food is the same as the pepper that you put on your food. And the ability to analyze our food just with a photograph and AI has gotten better and better, and yet it's still imperfect and imprecise. So I have no doubt that diet is absolutely fundamentally important in disease, but in terms of how to advise people with regard to their diet, that's a much more complicated issue. It may also be that the diet that's best for preventing one type of disease could trigger a different type of disease, or the diet that's best for one individual isn't necessarily the diet that's best for another individual. I think arguably the best data that I've seen about diet and disease comes from Stanley Hazen, who's a cardiologist at the Cleveland Clinic. And he's written about trimethylamine oxides that are induced by a carnivorous, a meat-containing diet, and are virtually absent in a vegetarian diet. And he's shown that TMAOs are very atherogenic. And so he's sort of led scientific support to the concept that a vegetarian diet is better in preventing atherosclerosis. You and I have talked about a fermented food diet, and a group at Stanford had a very insightful paper, I think, in Cell about 3 years ago on that, reducing cytokines. And I've had, at least anecdotally, one individual in my practice who thinks that her recurrent episodes of uveitis are markedly diminished by eating 6 portions of kimchi per day. But I don't know how universal that's going to be.
Right. Yep. Thank you. I saw a question from Chris, who put his or her hands up.
Oh, sorry.
Would you want to voice out directly?
Yeah. Hi. Hi. How are you, Dr. Rosenbaum? Here's Chris Muccioli from Brazil.
Oh, thank you for joining us, Chris. It's good to see you.
Yeah. Wonderful talk. And I have some quick questions, like very
Yes, you can
practical
just write down.
questions. What's your opinion for checking for microbiome exam for uveitis patients, as well as for probiotic supplementation for uveitis patients? Like, I mean, in general, do you see any room for this exam and for this kind of supplementation?
Right.
What's
So,
your opinion?
yes. So I personally don't take any probiotics. In the US, the probiotics are not regulated by the Food and Drug Administration. So you can sell whatever you want as a probiotic. And how many of those bacteria are actually viable is anyone's guess. And if they're viable, how many get through this gastric acid? Not so many. So I think Jeff Gordon actually did some duodenal sampling after probiotics, and there was perhaps some change, but not a lot. And again, there's no quality control for the drug. So I am not a big advocate of probiotics. I think that diet may well be a better way, but again, a very crude way that has effects that we just don't fully understand. And in terms of checking the microbiome in our patients, I hope someday that we will have personalized and precision medicine that requires us to do that, and we have a basis to advise them from what we learn by doing that. But we're not there yet. So it's diet's important, and I just have to tell patients that we're not as smart as we'd like to be. Do both those pieces of advice fit with how you would manage someone in Brazil?
Well, we have now many companies in Brazil doing microbiome tests, as well as a lot of offers of probiotics for everything. Like, okay, if you want a good health, you have to take some pre and probiotics and stuff. So, but that's why, I'm a biggest fan of functional medicine, which says that food is medicine. So that's what you're saying, that it's much better to check your diet than to take whatever. But I think it's sorry to say that, but it's a way to doctors make money.
Yes.
Yeah.
Yes.
Microbiome tests or probiotics for everything. So everything now is dysbiosis. So, your gut is not well, so you have to take
Maybe
whatever.
Dr. Padma and Dr. Biswas also have a query. Dr. Padma first, maybe, sir. You want to first?
I just like to know that, are you ready to tell that HLA-B27 positive uveitis, that microbiome is an important now in your disease? When you tell this, the patient always asks that what is the remedy for it? So should we say that the patient asks the questions, why I am getting this recurrent uveitis? Then we can say that is the gut microbiome is one of the cause. And if are you ready to tell that to the patient, or because we don't know what's the treatment later on will suggest to them
Yeah.
if it's give this information.
So, first of all, I agree with Christina that there's a lot of quackery and exploitation involving the microbiome, and so we need to be very careful how we communicate, which also relates, Dr. Biswas, to your question. You know, I share information about the pathogenesis of the disease differentially with patients depending on what I perceive, perhaps incorrectly, but what I think is what the patient wants to know or is able to comprehend. But for some patients, I do share that I think that bacteria are causally related to the inflammation. But then I also have to share that, you know, yes, I'm confident that that's true, but I'm not confident about what to do about it. So,
Yeah.
I hope the next generation is going to be so much smarter. If I have the perspective of seeing what my dad did 50, 60 years ago and how he practiced rheumatology and how I practiced rheumatology. And it won't surprise me if 50, 60 years from now, people are going to be shaking their head and they're going to say, those jerks in 2024 were giving TNF inhibitors to patients with inflammation. What a stupid thing to do, because that changes the microbiome so much and it doesn't get to the underlying cause and it has so much risk. And, you know, we'll be smarter one day, right?
Yeah.
And then, yeah, Dr. Pema.
Excellent talk, Dr. J. And I would like to ask you regarding the role of Ozempic, the GLP-1 receptor antagonist role in the management of autoimmune uveitis.
Oh, you know, I mean, the GLP-1 drugs have been such a surprise in terms of how many applications they have. And they may well have an effect on inflammation. Certainly obesity has an effect on inflammation, so they'll have some indirect effect. I haven't seen data about GLP-1s and uveitis. Have you?
No, I just wanted to know because even though it's used for obesity that has more anti-inflammatory properties, so definitely it will find a role in the management of uveitis. So that's why I just wanted to ask your opinion on that.
Yeah, no, we've learned a lot about TNF by having TNF inhibitors. I'm sure we're going to learn a lot about obesity and GLP-1 by having drugs that are GLP-1 agonists. So, but I haven't seen good data yet on using those for inflammatory diseases.
Padma, we can do a study then together.
Yeah.
There we go.
Dr. Vishwas, we can do. Yeah.
There's a question from Deepa online. Deepa, are you here? Would you want to voice out?
Hi, good morning.
Good morning.
Yeah, good morning, sir. It was a nice talk. So this is my first time participation, and I'm working currently for Dr. Rupesh. So I was just basically I'm a palliative care counselor. So I was just interested in asking the question about in patients with chronic or recurring uveitis, how can palliative care approaches, including managing the microbiome, help improve comfort and reduce inflammation?
Well, at least in 2024, I don't think that we have a good approach to the microbiome for that patient population, but you can send the patient to Dr. Rupesh and he'll help you out.
Kajal, you want to ask?
Yes, good. Thank you so much, Prof. Prof, I just wanted to check with you the role of oral microbiome, especially the flagellated bacteria that we have, the pathogens, especially the P. gingivalis and Fusobacterium nucleatum. Because both of them can be detected by toll-like receptors 5. So do we think there could be some commonalities between these bacteria and the gut bacteria?
So it's a great question. In rheumatoid arthritis, a lot of the interest has focused on the oral microbiome, and there is some correlation between gingival disease and getting rheumatoid arthritis. And we have looked a little bit at the oral microbiome. The skin microbiome, both in atopic dermatitis and psoriasis, is presumably important. And of course, every inch of skin is a little different ecosystem, right? So the bacteria on the crease of my nose are different from, the bacteria that are here in my chin. And when you're studying the oral microbiome, the depth that you sample from the gum and which tooth you sample, it's all complicated. So I think that the oral microbiome could be related, but I think the story with diarrhea-related diseases and the gut is more cogent for me right now. But I'd love for someone to prove me wrong.
Thank you so much.
Tony.
Yeah, thank you. I think time is about up, so I think we do have to close the section. But before that, maybe put our hands together again for James. It has been very insightful and a wonderful talk.
Maybe a group photo, if possible. We can switch on the video. And the colleagues from China can come into the center of the video.
Okay, so we do a virtual photo.
Virtual photo.
Okay.
Okay.
There
Thank
we
you.
go.
Thank
Great.
you so much.
Nice memory for me.
Thank you so much.
Thank
Okay,
you.
dah. Okay.
Wonderful.